Okay. Hi, everyone. So we are going to continue thinking about some aspects of cells and organs of the immune system today, and then we'll move into thinking about the humoral innate immune response, and I will, of course, define what I mean by humoral when we get there. We left off last time thinking about organs of the immune system. I had specifically told you about the primary lymphoid organs, which are shown here in red, the thymus and the bone marrow, where immune cells develop. And we had started talking a little bit about the secondary lymphoid organs. And in helping to understand the secondary lymphoid organs, I had introduced a problem that we see in the circulatory system, specifically the fact that there could be this issue with pressure in the circulatory system. The way that that is dealt with is that we have fluid that actually comes out of the walls of the arteries. The walls of the arteries are permeable to liquid. And so we have fluid coming out of the arteries, which is good because it relieves the pressure problem. It's also good because then all of your cells are bathed in nutrients and other kinds of fluids. So it sort of is serving a couple nice purposes there. But we also have the problem that this fluid needs to be collected back up so that we don't turn into a giant blob. So this fluid, which sometimes is called interstitial fluid, is recollected by this set of vessels known as lymphatic vessels that are part of the lymphatic system. I often refer to the liquid as the lymph because it's the liquid in the lymphatic vessels. So you may have heard of interstitial fluid. I'm pretty much always just going to say the lymph. So as you remember, we have all this fluid and we need to bring it back into the body, into the circulatory system. The circulatory system has the heart, this big giant pump to move everything around. The lymphatic system is set up a bit differently. The lymphatic system has lymphatic capillaries that are little and tiny, and then other lymphatic vessels. But we don't have a pump on this system. Instead, these are just vessels that are sitting open. And liquid can get into them if it happens to go by. All of these vessels have valves in them. You can see some of those valves here. And those valves make it so that liquid can only flow one way. So we've got these open vessels, liquid can come in, and once it's in, it sort of gets pushed in the direction we want it to go in. It can't go the other way because of the valves that are in the way. And so basically we're just collecting up any liquid that happens to be outside of cells in these little capillaries that are everywhere. As a bonus, it turns out that we can also pick up things like bacteria, viruses, or other things that happen to be in the body through these open vessels. And so we basically have our little lymphatic capillaries that join together to make bigger and bigger vessels, and we're sort of moving all of that lymph back to the circulatory system. The way that we move things through the lymphatic vessels, like I said, is not with a pump. So the way that we push everything the way we want it to go is by body movements. And so as you're moving different parts of your body, that's moving liquid in the tissues, making it flow by the lymphatic capillaries and into those capillaries. And so you can think about individuals who are bedridden, who often need movements, need to have their joints moved and things like that. And there are a couple of reasons, but one of them is actually to make sure that their lymph is moving and not pooling in their legs, say, or something like that. The really useful thing, in addition, about the lymphatic system is that at different places throughout this system, there are little specialized organs that do surveillance on this liquid that's going through. So basically we have little, I don't know, we'll imagine like little TSA stations that as the liquid is flowing through the body back into the circulatory system, we check it out to see if there happen to be any microbes that have been picked up in that area of the body. Those surveillance organs are the secondary lymphoid organs. So the secondary lymphoid organs are really the organs where we are searching for microbes, where we are trying to make an immune response happen. I've also sometimes heard the secondary lymphoid organs defined as the place where immune responses are initiated. They also, I have also referred to them myself as the place where the party happens. They're kind of the big important part of the immune response. I'm looking at the order of the upcoming slides, and I feel like the next slide is in the wrong place, so I'm going to just skip ahead for a second. And so this is really kind of how that circulatory process works. We've got the cells coming out of the heart in the artery. There are arteries and veins that are also going into those secondary lymphoid organs, so we can bring immune cells there that way. But we can also have this lymphatic that's bringing cells and liquid and eventually dumping it back into the heart. So finally we get all the liquid mixed back together in the blood back at the heart. You can see this happening here as well. We've got blood coming through arteries and veins, but we also have this lymph vessel that's going to be returning any of that liquid that was out between blood vessels into the heart. Specifically, it's usually brought into the thoracic duct. And so that may be returning cells. That is also going to be returning any lymph. And specifically, those are cells that may have decided they wanted to go to a secondary lymphoid organ. And you can see this again, just happening here. So we have these open-ended vessels that are collecting up liquid, but also microbes, cells, whatever from areas in the tissue, bringing them to a secondary lymphoid organ. We've also got arteries and veins at that secondary lymphoid organ, and we have lymphatic vessels that will help bring everything back to the blood. There are also these lymphatic vessels coming into the lymph node, and so there's sort of this circulatory system connecting all of those secondary lymphatic vessels. So if, for example, a cell decided it just wants to spend its life in the lymphatic system, maybe it's a certain type of immune cell that says, I'm just going to go around the lymph nodes and look for my bacteria of interest. It can just travel through the lymphatic vessels. It doesn't even have to go into the blood and just move around different secondary lymphoid organs. You have secondary lymphoid organs. Oh, sorry, Kyra.
So would you say that the lymphoid, or like the secondary lymphoid organ is kind of like a filter?
In some ways, I could think of the secondary lymphoid organ as sort of a filter. I can totally see that. So I'm going to explain one other piece, and then I'll sort of come back to another way that I could kind of see it as a filter. So one other piece about the secondary lymphoid organs is that you have a lot of them, and they're in many different anatomical locations. One of the most famous sort of sets of those secondary lymphoid organs are lymph nodes. You can see a general idea of a lymph node here. We've got an artery coming in, a vein coming out. We've got a lymph vessel coming in. We've got a lymph vessel coming out. And you can also notice that there are organized structures within that lymph node. You don't really need to worry about what those organized structures are at this point. They're easily visible under histology, things like that. But the other key piece here is that if you look at around the body, you will find lymph nodes sort of everywhere. So this green in this little person is the lymph nodes, and you can see these little is the lymphatic vessels. You can see these little kind of bean-shaped things at different places. Those are all different lymph nodes. This figure is a little bit out of date because we've actually learned that the lymphatics go up further into the brain, and we there's probably more going on up there than is actually shown in this figure. But you can basically see there are lymphatics everywhere. And each lymph node is really responsible for surveillance of a different section of your body. Basically, all the liquid and microbes and whatever else from that part of your body gets pushed through that lymph node. We often say that lymph node drains that part of your body. So, for example, if there is liquid or if there are bacteria in your sinus cavity, they all go through the lymph nodes that are right here on the way to the heart. So you've got lymph nodes here that drain kind of parts of your head and your sinus cavity. You've got a lymph node behind your knee that's draining the lower half of your leg. You've got a lymph node in your groin that's draining the higher half of your leg. You've got a lymph node in your armpit that's draining this part of your arm. And so there's sort of a lymph node for different parts of the body. And all of the liquid cells, potential pathogens, gets, as Kyra correctly says, filtered through that area. One of the reasons why that's really nice is that when we're going to talk about lymphocytes later on, we'll see that lymphocytes are very, very specific. We might say have a lymphocyte that is binding to a specific aspect of an influenza virus. Well, now let's imagine that that lymphocyte needs to go around the body and say, okay, you, cell in the very upper left corner of Dr. Barker's head, do you have influenza? Now, you cell next to it, do you have influenza? And if it had to go one by one through every cell of my body, by the time it checked every single cell, maybe like it checks this cell and keeps going and then influenza comes here, and it would be a while before it got all the way around and back up here. And by that time, influenza would have like totally taken me over, right? Instead, those lymphocytes can just hit the lymph nodes and say, is there anything in your area that's a problem? Is there anything in this area that's a problem? And then focus on doing further surveillance in only that area that was drained by the lymph node. So this system also allows for a lot of efficiency. In addition to the lymph nodes, there are some other secondary lymphoid organs. The most famous of those other secondary lymphoid organs is the spleen, which you can see here. It is in sort of the abdominal cavity. It's right in the back. If I actually look at the structure of the spleen, in a lot of parts, the structure looks identical to a lymph node. The only difference is that the spleen also has some other parts. So the white pulp is basically lymph nodes. The red pulp is for red blood cell recycling. So it's just the spleen is a lymph node that also does red blood cell recycling. And the spleen we often think of as kind of the lymph node that checks the blood. For a lot of immunologists, the spleen is also just a really nice source of immune cells. If I am wanting to use cells out of an animal, when I will dissect the animal, spleens are big and hard, easy to find, and like can get me 50 to 100 million cells, and it's just like no big deal, and suddenly I have all the cells I could ever want. And so, in my mind, sort of spleen and lymph nodes are sort of interchangeable. Yes, spleens do have another anatomic and physiologic job, but for us, they're really just the blood's lymph node. There are some other places where there are also secondary lymphoid organs, or secondary lymphoid tissue throughout the body. So there are some parts of your body that are often in contact with different microbes, and so there are sort of organized structures that look almost like mini lymph nodes there, to help do surveillance. One set of those little mini lymph nodes are found in the gut or the GI tract. And so we sometimes will refer to them as gut-associated lymphoid tissue, or GALT. This includes, most famously, these little bumps that are on the small intestine, which are like little mini lymph nodes. They're called Peyer's patches, which are basically little specialized pieces of the small intestine that are making almost like a mini lymph node. Because there are just so many microbes in association with the human GI tract. There are similar types of lymphoid tissues in other parts of the body besides the gut-associated lymphoid tissue. Sometimes I think the naming gets a little silly because sometimes in addition to gut-associated lymphoid tissue, we talk about MALT, which is mucosal. It's at mucosal sites. Or we'll talk about BALT, similar types of things in the bronchial tract, so it's bronchial associated lymphoid tissue. Sometimes we even talk about such tissue in the nose, nasal associated lymphoid tissue, or NALT. In every case, it's the same. It's sort of some organized structures that make almost like a mini lymph node in different types of body locations that are in contact with microbes a lot. And this is just showing you some examples of gut-associated lymphoid tissue, the adenoid and the tonsil, that also can function as sort of mini types of lymph nodes. So that's kind of the definition part for secondary lymphoid organs. I've also given you a definition for primary lymphoid organs. And so we finally have to think about this last type of lymphoid organ, which is the tertiary lymphoid organ. And so I'm going to give you a definition for tertiary lymphoid organs, and then I want you to give me names of organs that you think fit this definition, okay? So a tertiary lymphoid organ is a place where an immune response can happen. And so it's a place where you're actively having an immune response. So what are some organs that might be a tertiary lymphoid organ? Well, what are our examples of tertiary lymphoid organs? Where you might have a microbe to respond to. Yeah, Michaela. Okay, so skin could be. Yep, Grace. Lungs could be. Yep. Stomach could be. Yep. Honestly, it's every single organ. Any organ could be a tertiary lymphoid organ. So I guess I will sort of explain it that like right now, this patch of my skin is like fine. Everything's all good. So we probably wouldn't say this was a tertiary lymphoid organ, but I slipped and fell and like skinned my foot and have this really bad like rash thing on my foot. And so that skin is a tertiary lymphoid organ because I'm making an immune response right now in this little part of my skin on my foot. And so it's kind of where you have a tertiary lymphoid organ at any given time will change. It's wherever you've got something going on. And so, you know, this way basically I can take credit for the entire body. So if you looked at the title slide from today, the title slide talks about today, in fact, the syllabus talks about today being the day that we talk about humoral innate immunity. So I'm going to move into thinking about humoral innate immunity. If you look at the course schedule, we're talking about humoral innate immunity today. We're talking about aspects of cellular innate immunity on Wednesday and Friday next week. And then the Monday after, we're going to be discussing a paper that's related to cellular innate immunity. So we're going to be in innate immunity world for a while. I will send you, I usually want to make sure I have, when we have papers that we're going to discuss, that I give them to you a week in advance. So that paper will be on Moodle before Monday. Usually it would be by Monday's class, but we don't have class on Monday, so it will be up by Monday. When we're thinking about innate immunity for these next few classes, I want you to remember the importance of the rapid response. Again, if we had to just wait around for the slow adaptive immune response, we would always lose. because microbes reproduce just so fast. Also realize that this is the response that is relatively constant. It doesn't change from exposure to exposure with the microbe. And as you can see, this sort of mentions something about a limited number of specificities. It's relatively lower. When you are studying for exam 1, this point about limited number of specificities and sort of this point about fixed will make sense, because you'll be comparing and contrasting some things we've seen about some adaptive immune receptors. But right now, having not yet learned those receptors, it's hard for me to give you a definition that makes a whole lot of sense. For a really long time, and this was 100% true when I was in graduate school, a lot of immunologists spent most of their time thinking about adaptive immunity. And adaptive immunity is really cool. I did my PhD thinking about adaptive immunity. If you look at our syllabus, the number of days we spend on adaptive immunity is a lot more than the number of days we spend on innate immunity. When I was in graduate school, we'd all be like, oh, why do we even have to tell people about the innate immune system? Why do we have to TA this class where we have, we would draw straws about which TA had to cover innate immunity, because we all thought it was so boring. Everyone was like, this is the worst, most boring thing ever. It's like this rapid but constant thing. No one cares. It's so boring. Well, I want to just tell you quickly why you should care about innate immunity, this topic that we're going to be talking about for the next few lectures. I will also tell you that in the field in general, innate immunity is a very, very hot area. Some of this will come up as well when we talk about cellular innate immunity. The Nobel Prize in 2011 was given for some really key discoveries in innate immunity, sort of the area where everyone works, and is now what my lab works on. So now I sort of look back at my grad student self and I'm like, innate immunity, really important. You should not undervalue it. But I think this is one key way to understand that importance. In this graph, you are looking at time on the x-axis and amount of a particular microbe on the y-axis. And we can start by looking at the yellow line, which is kind of the idea of what should happen in a normal individual. You can see that the microbe infects the person, and that microbe reproduces, potentially exponentially, up until some peak level. And eventually, we stop that reproduction. We don't let that microbe keep reproducing exponentially anymore. We stop it at this peak. And then we control and clear that microbe and get back to basically zero of that microbe. That's what should happen. There are a number of patients who have been described who are missing an adaptive immune system. The most famous of them have a disease called SCID, severe combined immunodeficiency, and they are missing some enzymes that we'll talk about a little bit later. The green line kind of indicates what happens in SCID patients, those patients who do not have adaptive immune responses. What you can see is that the microbes, again, reproduce pretty quickly, and they sort of eventually stop at some peak level, but they never get cleared. We sort of never have the ability of the immune system to get rid of the microbe. We can kind of slow it down, but we can only just kind of contain it. We can't actually fully get rid of that immune system.
I didn't get that. Could you?
Man, Siri. The red line shows what would happen with an innate immune deficiency. The red line is hypothetical. We have never found a person who doesn't have an innate immune response. It seems as though not having an innate immune response is incompatible with being alive. So you can't be alive without an innate immune response. So that should already tell you it's probably kind of important. We can find people who don't have adaptive immune systems. We can't find people who don't have innate immune systems. They die immediately at birth. And what would happen without an innate immune response is that that microbe would come in and it would reproduce exponentially. You can see it's reproducing even faster than in the other cases. And it would basically never get stopped and never even get stopped at some nice peak level. And so what you should realize is that even though some parts of the innate immune system don't have the bells and whistles that people get excited about with adaptive immunity, you wouldn't be alive without it. So it's kind of important to understand. Past immunologists got really, really into this distinction between cellular and humoral immunity. Personally, I don't love it, but I think there are some places where the distinction works for us. And so these are actually, well, this is a picture of one of the immunologists who is very involved in this debate. But these past immunologists, we're talking like 1900 time, argued, is the immune system cells or is the immune system liquid? And specifically, they meant like the liquid of the blood. Were the cells the important part, or was the liquid the important part? And so some people said, well, it's the cells. And some people said, it's the liquid. And they were thinking about like the 4 humors. So they were saying a humor is important. There were experiments that supported each claim. The answer in reality is both. But sometimes when we think about different aspects of the immune system, we talk about, well, this is cellular or this is humoral. And as I'm talking about innate immunity, this is one place where the division really works for me to divide this into the humoral parts and the cellular parts. But there's also one place where this distinction can get, well, there are a couple places this distinction can get confusing. When we talk about something being humoral, the late 1800s, early 1900 immunologists were thinking about, magic liquid. We now know it's not magic liquid. It's actually proteins that are in that liquid. So really, whenever an immunologist talks about humoral immunity, they mean a protein that was secreted from a cell. And so really, we're talking about, are there proteins that are having this key effect? And specifically proteins that are secreted from the cell and away from the cell are doing their own thing? Or is it the actual cell? I got so confused about this as an undergraduate. So confused. Because guess where the proteins come from? They come from cells. And so I could not get my head to work about why we were talking about the humoral things when they came from cells. It really bugged me. But the idea here is the protein the thing that's really dealing with the microbe, or is the cell the thing that's really dealing with the microbe? And so today, for the rest of the time, we are going to talk about some key proteins that work in the innate immune system. So it's the humoral innate immune part of the response. And we'll do the cellular part next week. One of the reasons why it's important to think about the innate immune proteins sort of a little bit first here and separately here is that they are unique in terms of their timing when they act. Innate immune proteins act immediately upon a microbe coming in contact with your body. This is part of an image from your textbook. It sort of shows about some steps, and I'm just showing you the top part of the steps right now, where we have these innate immune proteins, and you can see they're acting in the first 4 hours. So even the innate immune cells actually take a little while to get their acts together. The very, very, very beginning, the proteins are what you got going on. And what's great about the proteins is that they are preformed. Right now, those proteins are in your blood and in your stomach and in different parts of your body. So if a microbe shows up, those proteins act at, times 0 seconds against those microbes. They need no prep. And so these preformed proteins can start acting immediately. In general, these preformed proteins are also really working against pathogens that are outside of cells, extracellular pathogens. They don't do much against intracellular pathogens, but they are going to be really useful against extracellular pathogens. Yep.
When you say immediately, it's that 0 to 4 hours you're talking about the first one?
Yeah, but really I would think about it as like from 0 to 4 hours, they're what you got. But they start at 0 hours. They are not waiting at all. No, they don't wait till 4, but from 0 to 4 hours, they're the only ones doing anything, is the idea. One other thing that's really interesting, about some of the innate immune proteins is that there are versions of these innate immune proteins in basically every multicellular organism. So as we get more complicated, particularly as we get to, say, adaptive immune responses, we're going to be thinking about things that are only present in certain types of organisms. Pretty much every multicellular organism I know of has innate immune proteins like this. Before I came to Drew, I was in another school, and there was somebody in the chemistry department who just worked on this protein that they thought was, like a cool chemical protein. And I'm like, right, it was a fish innate immune protein. And I'm like, you're doing immunology here, you realize that? And they're like, no, why would we think about immunology? And I'm like, yeah, it's in a fish, and it's just one of these in a fish. And another way that sometimes we talk about these proteins as that they are antimicrobial peptides. So they are proteins that are specifically acting and doing something against microbes. This is one of those places where I mentioned before our distinctions between barrier organs and innate immune response can get are not always clear. This is a place where things get a little squishy. Because you find the highest amounts of these antimicrobial peptides at the barrier organs. And they're present immediately. So were they a chemical defense, or are they part of the innate immune system? And I think people could argue it either way. But you can see antimicrobial peptides are key features that we see at the skin, the gut, the lungs, the eyes, the nose, the oral cavity. We've got antimicrobial peptides at all of those barrier organs, largely to knock down the numbers of microbes. These are some of the antimicrobial peptides that exist in humans. What I imagine that you can see here from just this slide is that there are a bunch of them. And this is just some of them. I'm not going to list every single antimicrobial peptide in humans for you. I'm going to hit a few highlights of really important ones, or point out a few places where we see some kind of key things going on with some of these proteins. In this table, you can see that we have a list of the location where each of these different proteins is found. And you'll notice that many of them are in our barrier tissues. And we also have a list of the specific antimicrobial activity of these different proteins. So, one thing that I took out this one slide because I decided it was really not worth me spending like 10 tons of time on. But I will just tell you that among the antimicrobial peptides that exist, there are differences in specificity. So some of them will hit like all bacteria, and some might only hit like gram-positives or gram-negatives. And I think that many of my examples are pretty broad examples. And so I want you to know that they aren't all as broad as these examples. But I didn't feel like I wanted to spend time on another example just to show you it didn't have to be broad. I will also point out that some of them can do things that maybe you wouldn't necessarily have thought about as interesting for defense, but they're super cool. So one of them I just feel like I want to call out because it's cool is lactoferrin. So it turns out that in order to grow, either inside of you or anywhere else, bacteria need nutrients. And one of the nutrients they particularly need is iron. Lactoferrin in your body is an iron binding protein. It just hides all your iron. So the microbes can't have it. And they can't grow very well. So simply doing things like binding and sequestering the nutrients so the microbes can't find it is a pretty nice way of limiting microbial growth. The more I've learned about some aspects of this, apparently like the battle for iron in our cells is like a crazy battle with microbes. It's called iron piracy. One of the other really famous antimicrobial peptides is an antimicrobial peptide known as Lysozyme. And I'm going to tell you specifically about Lysozyme. I want you to look closely at the word lysozyme and note that the end is Z-Y-M-E, like enzyme. I point that out because later in the semester, we are going to talk about an organelle in the cell called the lysosome, which ends S-O-M-E like an organelle. And you should not confuse those two things. Lysozyme, as you can see here, is an enzyme. And that enzyme cuts a specific type of biochemical linkage, which is known as the beta-1,4 linkage, that's found in a molecule called peptidoglycan. And peptidoglycan is found in the cell wall of prokaryotic cells. Different amounts of it in gram-positive or in gram-negative bacteria, but it's this purple and blue. It's actually in both. And lysozyme just cuts the bond in that and breaks cell walls of prokaryotic cells. So it will hit any prokaryotic cell while your cells don't have this. And so your cells are completely safe. You find large amounts of lysozyme in saliva and in tears. You also can find it in a number of other mucosal secretions, but in fact the best places to get it from are from saliva and tears. And so again, you can imagine parts of your body that are exposed to microbes where we're going to want to keep knocking down numbers quite a bit. One of the other big types of antimicrobial peptides is the defensin. Yes, sorry. No problem. Yeah.
So does that break down cell walls of the bacteria as well, or does it target?
It could. It's just targeting all types of peptidoglycan, and so it could target some of the good bacteria as well. Some of the ways that that our immune system distinguishes good from bad, we don't totally understand. But you could imagine that even the good ones you don't want to have growing to infinity. And so keeping their numbers lower is still probably something that's really important. So we also have this large class of antimicrobial peptides that are known as defensins. Again, I'm not going to go through every single defensin that exists. There are two things I want to point out here on this table that talks about defensins. And then I will tell you broadly about defensin activity on the next slide. So one of the things that I want you to notice about all of these different defensins that we see is that many of them are made in places like epithelial cells, and most famously, particularly Paneth cells, which are cells in the intestine. So if you look at your small intestine, you've got sort of this wavy area where you were trying to get a lot of surface area. And right at the bottom of all the waves, we've got some Paneth cells. And those cells in your intestine are making a lot of defensins. So again, we're seeing these made at high levels in those barrier organs. You will also notice that if we look at this part of the table about the regulation of synthesis of these defensins, that most of them are listed as being constitutive, which means on all the time, made all the time, made by all the cells all the time. And that's a key feature of all innate immune proteins, is that we're making them all the time so that they are there at time zero, ready to go and defend against different microbes. This figure from your textbook on the left shows how defensins work. I, as a student, found some parts of this confusing for many years, and so I have a little explainer on the right as well. The basic idea of how defensins work is that they are small charged peptides. You can see they have a positive charge on them. And that small charged peptide can insert into a plasma membrane and make a pore in that plasma membrane. So basically, defensins poke holes in plasma membranes. And then, the things that are supposed to stay in the cell leak out, and the things that are supposed to stay out of the cell leak in. Pretty easy to see how that works against microbes. The thing that sometimes can be confusing with defensins is that you might look at this and be like, but my cells have a plasma membrane. How is it that defensins don't destroy our cells if they're making them all the time? How is it they're only acting against microbes? And the charge on those defensins is quite important here. It turns out that if we look at eukaryotic cells tend to organize their phospholipids and put charged phospholipids, particularly those with a negative charge, on the inside of the plasma membrane. There's lots of reasons why that's good for eukaryotic cell biology. But the point is organization. Prokaryotic cells don't go through all that work of organization. They just put whichever phospholipids wherever. And so there are negatively charged phospholipids on both the outside and the inside. That means that there is something for that positive peptide to bind on the prokaryotic cell membrane. And so the prokaryotic cell membrane can bind a defensin and have a pore formed, while a eukaryotic membrane will not. And so oftentimes people are like, defensins, they poke holes in plasma membranes. But one of the things that is going to come up over and over with a lot of things we talk about this semester is, how do we make sure this thing destroys microbes and not our cells? And so I wanted to make it clear how a defensin could hurt a microbial cell and not target our cells. This is similar to something I mentioned earlier, but sometimes antimicrobial peptides will do things like block sort of microbial growth and not actually kill the microbes, like I showed you with lactoferrin. There are also times where antimicrobial peptides actually don't touch the microbe at all and just touch whatever the microbe uses to cause disease. So there's a lot of microbes that make proteases that are important for their life cycle, but those proteases are also the thing that causes us a problem. And so there are some antimicrobial peptides, like this one here, alpha-2 macroglobulin, that is basically a protease bait. And it's just a protein that has a site to bind up any of those proteases. When a protease binds, alpha-2 microglobulin changes its conformation and traps the protease. And is now like, haha, you can't go hurt the rest of the organism. I have trapped you. And so the microbe doesn't die. The microbe keeps on living. We haven't targeted the microbe at all. But the thing that's actually going to cause damage, we've trapped and inactivated. And so this is we've got a lot of strategies with a lot of different antimicrobial peptides that may block microbes from actually causing potential damage. Okay. if someone who knew a lot of immunology looked at the syllabus or looked at the title slide today and they saw the words humoral innate immunity, they would say, oh, I know what she's doing. Because there's this one other really important group of proteins in the innate immune system that are really famous. And as soon as an immunologist saw humoral innate immunity, they'd be like, oh, that's what she's talking about that day. And this set of proteins are known as the complement proteins. And so for the rest of the time today, I'm going to talk about complement. I am also probably not going to finish, and we'll talk a little bit about the rest of it on Wednesday after break. Particularly when I was back in grad school and we would hear about complement, this would really be where our eyes would roll into the back of our heads. We'd be like, oh my gosh, why do we have to talk about this? Again, it was described in like 1900. It was just really, really, really old. And some aspects of the biology haven't changed since then. In some ways, I think we used to think of it as like that old dusty thing that no one cares about. However, we are finding more and more and more places where complement is being involved in diseases that we never would have said were related to the immune system. And we're suddenly finding, oh wait, complement is the cause there. A lot of the problems we see, for example, in osteoarthritis, which is the type of arthritis you get just because you're old. Osteoarthritis is largely complement driven. This is actually an eye from someone with acute macular degeneration. And those are all complement deposits that are actually hurting this person's vision. And again, this is something that we would have been like, that's just a disease that just happens to some people, not an immune-related disease. And yet we find complement being part of the cause. And so more and more, I think now people are suddenly realizing a lot of places where complement is important, where they did not necessarily think of it as being important before. When we think about complement, a trick with complement is that it was described a really long time ago. And as a result, some of the parts have wacky names. And so some of the things you're going to look at and be like, what are these words she's saying? Because these are just the weirdest names. And it's because they were described such a long time ago. This is the broadest thing I want to tell you about complement. So first of all, sometimes immunologists abbreviate complement as C prime. So I've got C prime there. If I ever by mistake am just writing on the board or something and write C prime, I mean complement. Complement involves a bunch of different complement proteins. This blue oval is depicting one of those complement proteins. Most of them, but not all of them, have names that are like C and a number. This one's C3, like complement protein number 3. And these are just proteins that are secreted by cells that are floating around your blood and around outside your tissues and wherever right now. So you have a ton of C3 in your body right now. And when complement starts happening, a complement protein will get cleaved. There'll be some kind of protease that cleaves the complement protein. And we'll get fragments made of that complement protein. And when we make those fragments, usually there'll be one that floats away and one that gets stuck to the microbe or whatever other surface this whole thing is happening on. So usually we'll take our one piece of a complement protein, we'll cut it with a protease, we'll get a little piece that floats away and a big piece that gets covalently attached to our microbe or whatever else we're doing. This is largely what we're going to see happen over and over again as we're talking about complement. We often talk about complement fixation. Complement fixation really means sticking complement to stuff. Or sometimes people will talk about it as just like starting the complement process because complement gets stuck to stuff when you start the complement process. So this is your textbook's view of complement. And complement intimidates a lot of immunology students. And so what I want to do is I want to tell you what you're looking at and what some key pieces are of what you're looking at, and how I'm planning to discuss those key pieces as we go through. Because I think that if you know what the big picture is and know where different parts are fitting into that big picture, it's not nearly as scary as people think it might be. So when I think about the complement cascade, I first think about how we start it, how we turn on the complement cascade. And I refer to that as the initiation step. So you know, there are 3 ways you can initiate complement. There are 3 ways you can turn that complement on. I'm going to tell you the names and like the details of the arrows as we go forward. But right now, no, there's 3 ways. And notice from the way that I've drawn this, they all seem to come together. So you can start in 3 different places, but no matter what, you get to the same part in the middle. Some places will call this the amplification phase. Some will just call it kind of the middle. There's a protein that's really important here. It's called C3. So sometimes you just say initiation is how you get to C3. Then once you have C3, there's 3 ways you can get rid of the microbe. Immunologists often refer to things like this as the effector functions. We love the term effector functions. It was a really long time that I spent studying the field because we use it so much that I found out that everyone else doesn't just know what that word means. I used it so much, I was just like, everyone knows this word. It's a very obvious word. No, I learned later. No, everyone doesn't know what that word is. The effector functions is how you have an effect. It's how you get rid of the microbe. So basically with complement, you've got 3 ways to start it. All 3 of those ways eventually get you to C3. And once you've got to C3, you've got 3 different ways to get rid of the microbe. And so you could use this way of getting to C3 and then this way of killing the microbe, or this one and then this one and this one and then this one. You can sort of mix and match, however. This, again, is the figure from your textbook that is showing this entire process. And it's showing it kind of in a word form. Oops. This is a version from a different textbook. It's showing the exact same thing, but it's showing us pictures. So now we're seeing pictures of the 3 types of initiation. A little box of the amplification, and here are they just put names of the effector functions at the bottom. So it's the same thing in pictures if you think about this in a more pictorial way instead of in a words way. I'm just giving you 2 options. What I'm going to do now is I'm going to start by walking us through one of the initiation phases. So we're going to do the whole arrow from the top until C3. Okay? After we've done the top until C3, we're going to talk about what happens after C3. So kind of the 3 options down here. Then we'll go back to the 2 other ways you could get to C3. Obviously, again, I'm not going to get through all of this today, but that's kind of the overall plan of where we're going to be. The first way to start the complement cascade that I want to tell you about is a way that's known as the classical pathway. The classical pathway was the first one we described. It's the first one immunologists figured out. I'm going to run out of height. So it's the classical pathway. We now know that the classical pathway is actually the last one that evolved. So it actually came in evolutionarily last. There are some organisms that can't do classical but can do the other ones, but it's the one most people like to learn first. And you'll see why. There's a reason why. So first we have to think about, okay, so how do we get this whole business starting in the first place? How do we start the classical pathway? And in the classical pathway, we start by having an antibody, which is this Y-shaped protein, binding to something on the surface of the pathogen. So you can see here, the classical pathway is initiated by antibody binding to a pathogen surface. So we start with antibody. That antibody can be in different types of geometries. We can have different types of shapes going on here. Right now, you care that it's the antibodies binding. A reason why this is really important about the classical pathway, this is a thing to like put a little star next to. Antibodies are part of the adaptive immune response, which means this is actually a way that the adaptive and innate immune responses come together or are integrated together. The other two ways that we're going to start the complement cascade don't need adaptive immunity. So organisms that don't have adaptive immune responses, they can still do those. But classical, you got to have antibodies. So you actually have to have adaptive immunity as a part of this one too. Once those antibodies have bound to our pathogen surface, you can see they have these little tails that are sticking up. And we are going to have a protein that binds to those little tails. Now, if you were in charge of the world and you were going to name this protein the first complement protein that does something in the complement in this cascade, what would you name this protein? I told you a little bit about how we name complement proteins earlier. What might you name this one? Yeah. C1, perhaps? So the first protein that acts is C1. That's why we usually start with the classical pathway, because it starts with C1. This is a cartoon image of C1 on the left, and this is actually an electron microscopy image of C1 on the right. It really does look exactly like that. And we usually talk about it, say that it looks like an upside down bouquet of flowers. And so it has this very specific geometry. And that geometry allows it to bind to those antibody tails that are on the surface of the pathogen. So C1 binds to the antibody on the surface of the pathogen. I'm oversimplifying some bits of C1 here because for our purposes, we don't need to do all the biochemistry of C1. I'm just going to say when this binding happens, C1 has a conformational change. There's actually a lot more to it than that. Conformational change. Magic. When C1 has its conformational change, it becomes a protease, an active protease. As a result, C1 now has the ability to cut the next complement protein, the next one in the series. So now C1 is a protease. And it turns out we learn with the classical pathway that immunologists don't know how to count. Because the protein, so here you can see C1 binding to those antibodies. And C1 is going to cut first C4 and then C2. So basically what happens is we cut C4. The little part of C4 floats away. We never are going to talk about it ever again. No one cares. But the big part is going to get stuck to the microbe. When the big part gets stuck to the microbe, it is, oh my gosh, an active protease. And its ability is to cut C2. And now we leave a piece of C2 stuck to the microbe and another piece floats away. The way that we name C2 is really weird in terms of the parts, whether they're A or B. So I'm not even going to focus on that. I'm just going to say we cut C2. So you can see we go antibody, C1, 4, 2. From what you've seen so far of the complement cascade and what I've told you about the complement cascade, what do you imagine happens when you have this piece of C4 and piece of C2 sitting on the surface? What do you think this combined thing can do? Yeah. Okay, it's a marker.
It's kind of like, hey, over here we have
Yeah, so it is a marker, but it has another thing it can do too. It has yet another function. What if I said instead of function, it has an enzymatic activity? What kind of enzymatic activity might it be? What'd you guess? Yeah, Grace.
Does it cut C3?
It's a protease. And the thing it cuts is C3. So we're done. We just did the whole classical pathway. Antibody, C1, 4, 2, 3. That's it. And so you can see sometimes people call it the C3 convertase because it cuts or converts C3 because we named this a bazillion years ago. And here you can see, oh my gosh, now we're cutting C3. You can see that we make this little piece C3A, who we will eventually care about, but we also leave C3B attached to the surface. Okay, so we did the whole top half of classical. That was it. So again, people get super freaked out by this. It's not that bad. Okay, Grace.
Which one cuts C2?
C4. Yeah. So that's basically what's going on. Once we have C3 cleaved and on the surface of the microbe, we're now here. You can see we got C3. We're ready to go to that bottom part. C3 always, no matter which thing we're doing, always has one thing it does that's the same. And that is that it cuts C5. Can't be 4 because we already did 4. So it's got to be 5. And so there are some people who say the middle is C3 and C5. Not getting super stressed about that. But C3 and C5 are kind of working together and they make all these other branches happen. I'm going to tell you one of those other branches right now. And then we'll pick up with the rest of this on Wednesday. One of the other branches is easiest for us to know numbers-wise. It involves a bunch of proteins. 4 of them, in fact. Well, some of them are there multiple times, but 4 different proteins. They're C6, C7, C8, and C9. And you can see their actions in this figure from your textbook here. Once we've got C5 active, C5 is cleaved into a big piece, C5b, and a small piece, C5a. We will care about C5a again, just like we care about C3a again on next week. But once C3b is around, it cleaves all the other numbers. We get a whole bunch of C9, and C9 makes pores and membranes. So whatever membrane this was getting attached to, we now make a giant pore in, and we call that the membrane attack complex, or the MAC. And this is actually membrane attack complexes in bacteria. Here you can actually see the side on of this pore on a cell. And in the end, we basically are just lysing the bacteria by attacking the membrane or making the membrane attack complex. So one of the things that complement can do is membrane attack. Yes, Alexis.
Does the C5 cleave those numbers in numerical?
It does, yeah.
Okay, so it's not like 1 to 4, 2 to 3.
No, basically when you get to, once you know C5b, I care that you know like 6 to 9.
Okay.
So yeah, I don't really stress about specific details. And 9 is really the poor one. There's a lot of 9. So this is one of the ways that we can cleave or we can kill bacteria just using proteins. We didn't even need a cell to kill bacteria. Well, I mean, we needed a cell to make the proteins, but minor detail. There are 2 other ways that we can get rid of microbes using the complement cascade. A couple other ways we can start. And so that's what we'll start with on Wednesday. Have a great weekend.